Hi everyone! Thank you for clicking on my video. Today I'm going to introduce an experiment on lactase and lactose intolerance. 12.4 pounds. That's how much US dairy consumption per person increased in 2021. Data from the USDA Economic Research Service reports the new record of 667 pounds of dairy consumed per capita in the United States. From this, it can be seen that dairy products are very important for humans. Dairy products contain a large amount of lactose. Lactose is a disaccharide molecule that once ingested by the human body needs lactase secreted by the villi of the small intestine to break it down into galactose and glucose monomers through the hydrolysis, allowing it to be absorbed and digested by the small intestine cells. Now, let's move on to the next part to focus on the experiment. We will measure the lactase activity under different reaction times using spectrophotometry, which requires either the substrate or the product of the reaction to absorb light in a visible light spectrum. However, none of lactose or galactose and glucose do. Therefore, we're going to use another molecule with beta-galactosidic linkage that has a similar structure as lactose and can be hydrolyzed by lactase, orthonitrophenol beta-degalactopyranoside, or ONPG for short. When hydrolyzed, its product, orthonitrophenol, absorbs light within the UV spectrum, so we can use a UV spectrophotometer to determine lactase activity. Now let's start the experiment. Crush one commercially available lactase tablet containing 200 mg of the enzyme to an even powder using a mortar and pestle. Place the resulting powder on a diagonally folded wetting paper, which functions as a funnel to transfer the powder easily. Then transfer the powder to a 50 mm tube and label it suspension. Add 10 mm 100 millimolar of phosphate buffered saline to extract the enzyme. To maximize the extraction, vortex the tube for 1 minute. Pipette 1 ml of the lactic suspension into two 1.5 ml centrifuge tubes respectively, and label it as lactase suspension. Set the centrifuge at 10,000 RPM and centrifuge the two tubes for 1 minute. Pipette 500 microliters of the supernatant from each tube to a clean 1.5 ml tube to make 1 ml of lactase extract. Prepare 41.5 ml microcentrifuge tubes and label them as described in the table. Perform the corresponding operation on each test tube according to the table and immediately place it in a dry bath set as 37 degrees centigrade for the desired time. Each time point is conducted in triplicate. Please pay attention. When the time is up, immediately take the tube out and add 500 microliter of 1 molar sodium carbonate solution in the tube to terminate the reactions. Before spectrophotometry, we can see relatively clear experimental results. As the reaction time extends, the yellow color deepens, indicating an increase in product concentration. After using spectrophotometry to measure the absorbance of the reaction content in test tubes, we can create a bar graph using the average values. By conducting a student sample t-test, we can determine statistically significant differences between the samples. This indicates a positive correlation between the absorbance of the tested solution and the enzyme reaction type. Moving on to a scatter plot, the linear regression equation and the value of R squared also show a linear growth trend in the absorbance of the tested solution during this time period. While we may not know the exact concentration of the tested solution, according to Beer's law, concentration and absorbance are positively correlated. This means that the absorbance of the solution can represent changes in product concentration. Since absorbance correlates positively with reaction time, it indicates an increase in product concentration, implying an increasingly potent enzyme catalyzed reaction and more significant enzyme activity. However, most of the time, what we see are graphs showing the relationship between enzyme reaction rates and substrate concentrations. The graph looks like this. At point A on the graph, the rate of reaction is proportional to the substrate concentration. At point B, the rate of reaction starts to decrease. And at point C, the rate of reaction is constant. This is the point of saturation. Let's look at each point in more detail. Starting with point A. At low substrate concentration, the rate of reaction is proportional to the substrate concentration. This is because there are plenty of active sites available to bind to the substrate. For point B, as the substrate concentration increases, the rate of reaction decreases and is no longer proportional to the substrate concentration. This is because some of the active sites are occupied by substrates. Now let's move on to point C. At high substrate concentration, the rate of reaction is constant and independent of the substrate concentration. The enzyme is saturated with substrate. This is the point of saturation. Now, I would like to introduce a condition related to lactase that someone around me is experiencing: lactose intolerance. People with lactose intolerance don't make enough of the enzyme lactase, which is needed to digest lactose. As a result, they may have diarrhea, gas, and bloating after eating or drinking dairy products. Some people may wonder at their congenital or developmental lactose intolerance. It's possible, but uncommon. This disorder is passed from generation to generation in a pattern of inheritance, meaning that both the mother and the father must pass on the same gene variant for a child to be affected. If you happen to have lactose intolerance, you may wonder when to see a doctor. If you frequently have symptoms of lactose intolerance after eating dairy foods, particularly if you are worried about getting enough calcium, it should be suggested to make an appointment with your doctor. The treatment is usually lower the amount of lactose in your diet, such as limit milk and other dairy products. Include small servings of dairy products in your regular meals. Eat and drink lactose-reduced ice cream and milk. Add a liquid or powder lactase enzyme to milk to break down the lactose. Of course, reducing the dairy products doesn't mean you can't get enough calcium. People can still maintain good nutrition, since calcium can be found in many other foods, such as broccoli and leafy green vegetables, calcium-fortified products, canned salmon or sardines, milk substitutes, origins, almonds, brazil nuts, and dried beans. Now let's move on to the final part, the thank you note. Firstly, I would like to express my gratitude to my lovely professor, Junhong Lee. It was truly a delight to encounter a teacher in university who remembered all the students' names in the first week. Thank you for patiently addressing my questions and comforting me when I was concerned about my grades. Your dedication has been the primary motivation for me to earnestly study this course. I also want to thank my biology tutors, Yuxin Wang and Christina. Attending all the study group sessions proved immensely helpful for my revision outside of class. Additionally, my thanks go out to my ThinkPad group for their encouragement and innovative ideas during our discussions. Thank you very much for paying attention to my video. See you in the next lab.